Hello everyone, today I'm going to be talking to you about the process of decoding. So the process is from the DNA to the mRNA, which is messenger RNA, tRNA, which is transfer RNA, which will then lead us to the amino acids in each strand. So we're going to talk about what the DNA is made up of. It is made up of adenine, cytosine, guanine, and thymine. So in the DNA, adenine binds with thymine, and cytosine binds with guanine. Now when it comes to the mRNA, it is made up of adenine, cytosine, guanine, and uracil. So adenine will bind to the uracil and cytosine will bind to the guanine. The only difference between the mRNA and the DNA will be the uracil, which is the mRNA, and the thymine that is in the DNA. Now the first example, we're going to start off with the DNA strand, TAC, GCA, TGG, and AAT. So the complementary strand to the DNA will be whatever it binds to originally. So thymine binds to arginine. So A will bind to T, C will bind to G, G will bind to C, C will bind to G, A to T, T to A, GC, GC, AT, AT, TA. Now, the mRNA strand, remember, has uracil, so A will bind to U. This will be it. So A will bind to U, T will bind to A, G will bind to C, so forth, G, C, A, U, G, G, A, U. Now, in this example, the mRNA will take the strand out of the nucleus and into the ribosome. Then the tRNA will bond next to the mRNA. This is where you'll get U binds to A to U, C to G, then we have CGU, ACC, UUA. You will then use a tRNA strand to find the amino acids. So every 3 letters will make up what we call a codon, which carries a particular code for an amino acid. Therefore, this is a codon, this is another codon, this is another codon, this is the last codon in this strand. You will then use what we call an amino acid chart, which looks like this, to find every amino acid in the strand. So we will start with this first one, which would be AUG, which we will find here, which is methylene. You will continue to look for each codon on the amino acid chart, and the end result will be methionine, arginine, threonine, and leucine. Now, a little quick point, when it comes to mutation, if this letter were to change, but the end result would be arginine, then there is no mutation. But let's say this letter that changed ends up being, let's say, serine, then that would be a point mutation in this DNA strand. Thank you for watching. I hope this helped with coding.